Thanks, Rita. I'm just going to share my screen, and thank you to the audience for joining today. So let's just kick off with putting the slides in, and here we go. So dealing with protein mobility, we'll be talking about docking and induced fit docking using molecular mechanics. And we will cover a number of things that you already saw in the agenda if you registered on our website. And so to start off with, docking in GLIDE, if you've been using it before, assumes that the protein is rigid. So we perform conformational sampling, as you can see in the image below, on the ligand as it goes through the GLIDE filter, and we have different distinct ensemble of ligands, and we generate poses for them, and we dock to the receptor. So the receptor is shown here with a different shape to the different ligands that are coming in, and we try to orientate the ligand and sample the ligand relative to the receptor so that we get some form of receptor-ligand binding. So some things bind, they can bind, but they're not binding very well, but that doesn't mean to say that they're still not making key interactions. So the next part of the process is the scoring aspect, and here is how good, the question is how good is a given pose. So although all of these things bind to a rigid receptor, we have a score of roughly -11 to -5, although I have seen things outside this range. Arbitrary number that sort of resembles the free energy to give us a feeling for how well something binds in the shape complementarity and also the electrostatics and various other forces that are involved in thermodynamics of binding. So for example, here we see although it binds well, it doesn't bind as well and has a smaller score. So this is an example from a real screen that was used on the COX-2 in protein. So here, I think, yes, what we'll see is that the crystal structure is shown in the first image in blue, and we go ahead and as we screen the compounds, the hits, and have a look at the top ranking hits, we can see that actually 2 or 3 molecules right at the top already start to make the key interactions that we're looking for and resemble the way that the co-crystallized structure sits in the active site. So this is an example of docking more than 100,000 molecules into the 6-COX receptor, and we used a particular type of workflow to achieve this, but the standard precision docking is what you see here. But what you will notice is that the protein is always static in these images. So the 2-step process in using Schrödinger tool for docking is grid generation for defining your grid. So we place a sort of active site box around the co-crystallized ligand in the grid generation. And then we perform ligand docking. So we take our co-crystallized ligand, if that's the first thing we do, and then dock to that grid which emulates the receptor interactions. So if you are used to using GLIDE in a more automated way, you may have already used the virtual screening workflow. So this actually helps us to interact with some of the GUIs inside the GLIDE docking, but also other interfaces within the Schrödinger software. So for large and hierarchical jobs where we want to run high throughput docking, standard docking, and extra precision docking with multiple receptors, we use a wrapper called the virtual screening workflow. So this actually includes things like ligand filtering, ligand preparation, and docking, as well as post-docking methods. And it also has a restartability in the event of any problems happening. So in the context of this talk, I'd like to say that this is a very useful workflow for including multiple prepared receptors to the interface to perform ensemble docking. And this gives you multiple sort of images of the way that your protein can be flexible in the face of screening multiple ligands. So here's an image of the screenshot of the interface. You see the multiple tabs along the top here, so for input and preparation of things, filtering of things, preparing the receptors, adding multiple receptors, And this is an example of where we have multiple receptors added just simply using this add button here. It's not a particularly good example of factor 2A, but you can see that there's 3 added receptors here. So in this case, the audio paired ligands and will be run through each of these receptors, and the outputs will all be separate for these receptors and zipped up, so you can compare them, and it's not confusing as to which ligands dock to which receptors. So to consider a single snapshot of your protein is not the exact way really to perform docking, because the exact conformation of that protein that was crystallized by the crystallographer really depends on the conditions that were being used and the ligand that was bound to it. So proteins actually, as we all know, are much more dynamic than these pictures suggest. So in this example, the phenylalanine is actually in movement in this protein, but with the crystallography data, you might actually only capture the gray image of that phenylalanine and therefore define how that ligand is docked. So it's a very different image of the way that that gray ligand is docked to that gray phenylalanine compared to if that phenylalanine was actually moving out of the way and the ligand is actually stacking on top. So to emulate those kind of movements, we need to really consider the fact that things beyond the use of glide. So anything that remodels the protein is going to be more CPU intensive than glide docking and using a rigid protein. So it's always a good question to ask yourself, when do I need to employ any kind of protein flexibility and to what extent? So how do you detect flexibility in your protein in the first place? And the way that I do it is basically to, first of all, overlay as many crystal structures as I can. So if I have a family of proteins that I can download from the PDB, I'll overlay them just to get a feeling for where there's known variation. Also to color things up by B factors of the whole protein, and to understand how things move very close to the active site and outside the active site. So we'll talk a little bit about that later. And also, you may be further along in your project, so you know that there's a large ligand that fits into a pretty small site. It definitely, it doesn't look like that it can fit, but it should, because you know that it can, and therefore you need to make more room in the binding site. And finally, if you have experimental help in your project, if it's known to bind experimentally and you can't get it to dock with rigid glide docking, you know that there's something going on there. So this is an example of the HIV protease, a few tens of structures from the family which are overlaid to superimpose. So you can see the secondary structure superimposed as well. The binding site is also shown, and there is some degree of variation in some of the arginines that are close by, maybe towards the outside of the active site. But it's generally overlaid reasonably well, but you'd have to look closer at the active site to start figuring out what's going on. This is an example of the 1GVQ structure, and this is colored up not by atom type, but by B factors, and B factors tell us more information from the crystallography and in your PDB file of how much atoms move away from their average mean position. The more the atom moves, the less reliance we have or confidence we have in the fact that it sits in one place. So if it sits in one place, it has a low B factor and it's a dark blue color, which is often referred to as a cold color. And if it has a high B factor, which means it moves a lot, it's actually going to be a lighter shade of blue through to pink and through to red, and red is considered quite hot B factors. So B factors around 20 are quite high, compared to around 4, which is quite low. So you can see that around 4, very close to the active site, is something that we have a lot of confidence in. It does not tend to move so much, but we can see some residues a little bit further out where the numbers are around 20 plus, and we can just say that these would be ideal candidates for the areas of protein flexibility and trying to model that flexibility in. Just an aside, that if you are looking for the color schemes and the legends for this coloring scheme, you will find this under the Preferences tab in Maestro. If you type in color, you'll see a color scheme here, and in there all of these are drop-down options for the different coloring schemes. And if you click on, if you highlight the row and go to Edit, you'll be able to see the actual coloring scheme here. So you can see that these are lower B factors and around here, and then higher B factors as it goes towards the red region and the lighter region. Another way of thinking about or looking at your structure when you first download it from PDB is to consider atom occupancy and local flexibility of side chains. And when the crystallographer has information that there may be multiple conformations of either the ligand, but in this case we have three residues shown here, they will add that information into the PDB file. So this is where either there's not enough electron density there, or it's dubious in some way, and you will have an ALT entry in your PDB file where it has a number associated with it. So the number 1 is 100% occupancy, and the number 0 is 0% occupancy. So here we have the serine in this example shown as 0.8, which is about 80%. And this is the one that you're actually going to view inside the Maestro interface. So in this case, we see the valine, which is 74% occupancy, and we see that in the solid image here, which is highlighted with the trace. But you also see a ghost image in the background, and you simply have to go to next position and commit to that position if you want to consider that one. Making a choice of how to consider that one is a different thing. So, I mean, in this case, it actually looks like 74% of the time the solid image is the correct one. So it doesn't make too much sense to go to the alternate image. But if you think there's enough room for movement, and if the ligand that's binding in needs to be bigger and sit inside that pocket closer, or move that lysine out of the way a little bit, and that actually reflects that, then that's a possibility of wanting to use the ghost image here. So this is available inside the protein preparation wizard, and it comes up when you prepare the protein as one of the problems. So if you go to view problems, you'll see alternate positions showing in here. But it's also available inside Maestro itself. So if you download the PDB and you start to prepare it or look at it, you can just quickly toggle on alternate positions. And when you flash this on and off, it becomes really obvious over your system whether something has got ghost images in it. And then if you select the residue, right-click and go to Additional Edits and switch alternate positions, this allows you to go from the solid to the ghost image, and that's a way of committing to that alternate image. Let's just quickly switch to the demo of some of these things. So we have here 3 unaligned proteins, and if you want to first download and look at these, the first thing that I would do is either use Quick Align to align all of these proteins, and I spoke about this yesterday a little bit because it uses no input from the user and uses center of mass to align things. Something that gives you a bit more control is protein structure alignment. So if you forget the names, you will send the slides, and also you can just type the names into the search bar. So here you can just type alignment, for example, and this will actually help in trying to figure out where to go. So here we have either all residues can be aligned, or you can pick specific residues directly off the workspace. So here, for example, if I just say workspace included entries, go ahead and align them, it will give me a little bit more output than the quick align and show me how the secondary structure is used to align things and also give me some kind of alignment score. The smaller the better. It gives me an RMSD, but it's usually sufficient to go ahead and just put things into the workspace and have a look at them and see how well things have aligned. Another approach to do the same thing is to use a binding site alignment. So this gives you a different level of control, I would say. So binding site alignment, it can automatically detect the binding site 5 angstroms from the ligand, or you can specify which residue to align. But this one concentrates a bit more on the binding site. So if you have a family of proteins that are actually quite similar but only different in the binding site, you might want to look at the detailed view of the binding site, then you can use this one. But in all honesty, they are all very, very quick algorithms. So if you're not getting the results from one, you can use the other one very quickly to see which one is more appropriate. So that's an example of where the binding site, and you can see the atoms that we use to define the binding site and the alignment, which happens in the background. So the alpha carbons that were used. This is the HIV protease site. So another thing that you can do is download your PDB, prepare it, and then go ahead and color things by adding PDB temperature factor. So here we have PDB. We display all the atoms, and also you may want to label things in the same way. So if it's not available in here, you can go to Edit Custom Label. Apply Custom Label. Sorry, so in the WebEx, very odd things can happen with Maestro. And I lost my Edit Custom panel there, but it has thankfully remembered the fact that it's trying to apply the PDB labels on here. So you can see that very close to the active site, we've got dark blue colors and low numbers. So there's a high confidence that these actually sit where they're supposed to sit. And then further out, we can see some slightly lighter colors there. You see that, you see this leucine here, 275. That's a slightly lighter shade, and we've got some higher numbers associated with that. So that gives us an indication that that this is possibly something that is likely to move around. But when you start to do the alignment and the coloring factors together in conjunction, all of these tools help you to figure out how much things are moving. This is an example of where we have quite a few alternate conformations of our side chains. So if I just turn on my alternate positions, you can start to see there that it's flicking on and off around this region. You just turn things around just to see where they are. I can just see one there, so I zoom into that area and now you can start to see that ghost image. So if I make sure that I'm in atom selection mode in A mode, double click to my residue and I right click, I can go to additional edits and switch alternate position. So you see here that the ghost image is being switched over. So this is also all available through the preparation wizard. So if you go to protein preparation, you will actually see this in, once you start to prepare, and I won't do that to save time, but you will also see this in view problems and have the ability to commit or keep whatever you want to do. So to press on, you have to consider important things before running induced fit docking. What's the purpose of the part of the protein around my active site? Am I looking at the metal binding site or catalytic residues, which actually are unlikely to move that much? So if you have key hydrogen bonds to those areas, it's a good idea that they probably stay where they are. But things like activation loops and ancillary loops, or loops in general that vary between isoforms, they have a lot of scope for actually moving around in induced fit. we also have to consider how badly is the section of that protein in the way of the ligand that needs to be docked. So as with all things in Comp Chem, they are either the right tool for the job or they're not the right tool for the job, because they won't take into account that they don't sort of have the right level of theory. So larger, very large overlaps will need a different method to be applied in IFD, and smaller changes require a different method to be used. But there are instances where if a ligand really doesn't look like it's going to fit or requires another level of theory, for example, molecular dynamics, then induced fit docking, which relies still on molecular mechanics, is probably not going to find you a sensible answer. But it's an answer that you can probably check within the space of maybe 24 hours. So you can run it when you in the lab, when you at the end of the day, and then when you come home and you come back the next morning, you should find an answer. Whereas molecular dynamics doesn't normally fit into that category of timing. So incorporating flexibility, there are several methods to do this, and we can start off by looking at how glide interface would actually cater to this problem by using softened potential models. So these basically lower the penalties associated with the small clashes and mismatches that you get between a protein and a ligand in trying to get the protein into that, trying to get the ligand into that protein. Those softened potentials can be applied on the ligand, but also to the protein itself. Now, if you're looking to do a bit more movement, so for example, key residues, then you'd use the full induced fit docking protocol. And in all approaches, once you've got multiple pictures of your receptor, then you can go ahead and use the multiple virtual screening workflow to dock those multiple receptors into things. So P38 MAP kinase is an example of where you'd be doing that. So this is the glide interface for ligand docking. If you've ever noticed the section at the bottom, we're allowed to scale the van der Waals radii of some things. So it softens the potential for non-polar parts of the ligand, where you can scale the van der Waals radii of ligand atoms with partial atomic charges of less than the specified value here. So we go ahead and change this scaling factor, and by default it's set to 0.8, simply because we've tried and tested lots of scaling factors over our data set of ligands to dock over our proteins, and we found that having a default factor of 0.8, so you're just scaling back up to 80% of those, so 80% of it is left as it is, and you're only scaling back by 20%, then things actually run quite well, and you get good enrichment. So this is an example of where we have a very small active site. We have pyric acid inside its cavity. An example of where we've actually softened the potential forces the pyric acid to sit closer and more deeper into the active site. So standard docking leaves the pyric acid in the green image, a little bit of softening around the surrounding residues, so this is in the protein, to 0.6, and a standard ligand docking, so there's nothing, so the ligand remains at 0.8, but the receptor goes down to 0.6, allows us to push that ligand a little bit more into the active site. And if we soften the potential even further, both the ligand and the protein, it moves it around a bit, but you have to be careful because you're actually creating more and more room, so the ligand has more degrees of freedom to do whatever it wants. And then the observed pyric acid position here is shown in the cyan. So really, softening that potential helps, but it doesn't completely get an exact image of the crystal structure perfectly. If you want to see this in a demonstration, let's just close the current project, open up the project. You can actually see this. I just ran it with a few examples, but you can see here the active site in the green one. The green pyric acid is exactly for the standard docking in Glide, so that's the standard docking, and the scale docking comparatively is in yellow. So you can see that it sits much further down in the active site. And we actually needed to get it to sit a little bit lower in comparison to the crystal structure, so that's in the cyan color there, but it's doing a reasonably good job. So under Glide ligand docking, you can set that, that scaling factor here to 0.6, for example. And in addition, when you run those grid generation, you can also scale the factor here. So by default, nothing is scaled on the protein, but you can scale it back to about 0.8 and see how it goes. It's a little bit of a trial and error to see how things are behaving with the scaling factor. So I would just go ahead and just run a few examples to see how your system behaves. So an example of small residue movement, we've got here the 1 GBQ and a relatively large ligand, which is prednisone, and it's a steroid drug used in inflammatory diseases. But you see here that I've just highlighted it all up in PDB color first of all, B factor colors, and you can see that everything is pretty dark blue, very close to the core part of the ligand. We have the flavin compound here, but we have the prednisone there in lighter blue, so that has some tendency to move. But we see here that we've got a couple of arginines here that are flapping around in the lighter color, and they have higher B factors. So that's the first thing that I would do, and you can see here that when we go to dock an example like this, that the kind of movement that we're actually seeing is this movement of this arginine, which we were able to capture and move out of the way using induced fit docking. So you can see here that arginine is really moving between the crystal structure and the IFD posy. So what is IFD actually doing to get this kind of result? It's a protocol that provides a complete workflow linking both the protein conformation changes and the ligand docking conformation changes. So at the beginning of this talk, we were saying that the protein in Glide is completely rigid, more or less rigid. So we combine the fact that receptor has a different shape to the ligand that it's trying to dock, but we know that this ligand can dock for various reasons, as discussed. So we go ahead and try to dock that ligand irrespective of the fact that that shape is different for the protein. So in some way we have to open up that cavity a little bit, get the ligand to conformationally sample inside that cavity, make enough room for it to be able to explore its conformational space. And then once we've done that sensibly, we go ahead and refine the protein around those docked conformations of those ligands. So now we start to move the picture of the active site of that protein. So it's going from a square to a round shape, trying to accommodate that ligand as it does that. But it all does is try to do this sensibly within an energy window. So we're only capturing things at the top of that energy window, the best scoring things, either from this round and from this round. And in the end, once we've modeled the protein around the ligand, we then go ahead and redock that ligand to get a sensible number so that we can actually start to quantify things. So in this respect, what we've done is conformationally sampled the ligand and the protein at the same time so that they can accommodate one another. Now, this is an iterative process using molecular mechanics. In dynamics, it's using a different process and doing this sort of simultaneously. So it's one step before you would start to approach MD. Now, in Glide we use the Glide score, and in IFD we use the IFD score, which is a combination of the basically the ligand being rearranged and the protein being rearranged. So we have to blend those two things in together in some way. and that's the number that you see at the end. But clearly this is a slightly cruder measure, but again, it's molecular mechanics, so I would not I would not rely on the fact that you've got the first pose to look at this, but you look at multiple outputs from IFD to figure out which is the right one. So here we have, under tasks, you can find IFD, and each of the tabs along here reflects the process that we just talked about. It samples the ligand, it docks the conformations of the ligand, it then refines the protein around the ligand, and then redocks the conformations and outputs the best energy result. So what we see here along these tabs is an exact following of that protocol. And in some respects, you'll see exactly how it matches the glide interface. So if you're used to using glide, then you will find this quite familiar. So this is an example of CDK2, and to save time, I might I'll quickly go through the interface as well. But you can see here that the cyan receptor 1PXJ is used to dock a ligand that doesn't quite fit into the active site in the same way that this ligand fits into its crystal structure. So this actually goes quite deep into the pocket and sits either side of these lysines in this position. But when we come to dock a potent ligand for this cavity, actually it straddles the binding site and hits both of these lysines either side. So clearly, if this ligand docks, these two residues need to move out of the way. And you can actually see in the orange residues, you can see how much movement there is generally using the IFD. So let's quickly take a look at this and how to set things up. When you've got the surface generated, you can actually see how deep that original crystal ligand goes into the active site. But when you put in the orange ligand, you can see that it's actually a completely different pocket and sits right through that surface, cuts through it, and you can see it hitting both lysines on the other side. And what we're really trying to emulate is, if I remove the surface and put the crystal structure of the orange structure back in place, you can see how much movement there is either side of those two lysines to get that ligand in place. So with our input receptor and our ligand highlighted in the project table, we use induced fit docking. You can also search from it in here. So if you just search for docking, if you forget the name, you can still access that same panel, induced fit docking. And the ligands to be docked are in the selected entry from the table. We use a standard protocol. Unless you have many CPUs, then you can generate up to 80 poses for the ligand. But when you generate use standard, then you generate 20 poses for the ligand, which is still a very fair number to achieve what you want to do. So if I just remind you back of this protocol, the conformations that are being sampled here are hundreds, and then in the end you just pick up 20. If we just go back here, we define the center of the binding site using the workspace ligand. You can just pick that straight off, and you can see that that's actually already done, because I think it remembers it from the last time I was doing this. Again, it's the same sort of bounding boxes that define the binding site. But you can make those bigger and change the length of those if you think that it needs to be bigger. So that's when it's useful to actually just put your other ligand that you're looking to dock into the workspace at the same time and see that it sits inside the purple box. Constraints, if you have any. So for example, if you were putting in hydrogen bond or metal constraints, you would pick those directly off the workspace. And you may also want to not allow your core part of the molecule to move too much. So you could use sort of a smart pattern to say that, for example, I don't want my orange ligand to move too far away from this general space. So you can use a smart pattern of that ligand to define that space as a core constraint. The ligands that will be docked are sampled conformationally, docked into the receptor, And in this case, we trim the side chains either automatically through B factor or manually specifying those residues. Okay. So just going back to the image that we were looking at, I mentioned that in order to get the ligand to fit inside the site, we soften the potential, but in some cases we also trim back the side chains as well, because it's softening the potential even to 0.7 and 0.5 for the receptor and the ligand respectively is still not enough. So we go ahead and manually trim the side chains using specified residues here. So here I've basically picked the residue off the workspace, specified each of those lysines just by clicking on them here and saying OK. If we were to choose automatic, it uses B factors, but it does use other criteria as well. And finally, once we've got all of the conformations sampled inside that site that now has a bit more room, it then goes ahead and refines that site around those conformations of the ligand, 5 angstroms around them, and we can choose to keep additional residues completely still, or we can additionally add further residues in. So if there's more movement around some of the loops, for example, you may want to refine those. If there's, for example, metal ions, you may want to keep those completely still. So finally, we go ahead and redock things into an energy window, and we use SP to see the end output structures. So if you run this, the outputs will be always in the form of a complex. So it outputs it as a single group. You see here the protein and the ligand, and these are ordered by IFD score. So the best is at the top, and we can start to see the kind of movement that it was able to explore. So if you just put a few into the workspace, you can see that's one of the lysines here, and they're actually very steadily moving in the same way. And the other lysine is just here on the edge, and there's some variability in the way that sits. And we can compare that to our crystal structure of the orange crystal structure, just to see how well things are doing. In this case, actually, we've captured something pretty decent and overlays quite well in the first instance. But you can see here that the output in that energy window is up to 23 structures. So I would happily look at anything up to about the first 10 or 5 structures, just to see what looks plausible. And if you can't figure out which one it is, then you can take maybe a representative set of those, so 2 or 3, maybe 4, you can representatively dock into using ensemble docking in the virtual screening workflow. So you go back and dock into all of them. So now let's consider slightly larger loop movements as we draw to the end of this talk. This is an example of where we have a crystal structure of TACE in cyan. It's making a hydroxamic interaction to an ion here in this part of the molecule, and it's sort of traversed over the top of that active site here in the blue color. We have a very potent inhibitor of TACE sitting in orange here, and you can see that actually it sits in almost completely different pocket. It's actually the P1 pocket of TACE, and although it makes that clear interaction with the hydroxamic acid, the rest of it is actually causing parts of the protein to move out of the way in order to make this very potent interaction down here. But if we use the blue crystal structure to dock the orange ligand, we would not get GLIDE to actually dock, and possibly not even, indefinitely not even IFD, because there's more than just a couple of residues that are moving. We've got a whole loop movement here. So what we really need to do, you can see that if we use IFD with loop movement in the right way, we can actually cause that loop to completely move out of the way, allowing that ligand to dock. This is just another view of the whole thing, just to give you a feeling for how much that's moved. I think it's about 8 to 12 angstrom movement of that backbone. Now, one way to do this is to use prime loop refinement. And this will allow you to explore different conformations of that protein loop. You simply put the protein into the workspace, use prime refine loops, find loops in the workspace structure all, and then decide which loop it is that you want to sample. So this auto-populates all of the table here, and then you can just pick the one that you want and edit the output. So let's just have a quick look at how that would actually work in here in the interface. So in this image, you can actually see how that that NAT file grid is sitting so close to that loop here, it really needs to move out of the way. So we can use define loop and prime. Find all loops in the workspace. And it literally uses the PDB information to do that. And in this case, we would have to physically define and make a decision on what needs to be moved. So if I put the very potent selective ligand into the workspace, you can actually see and make a decision about what part of the loop really needs to move. So in this case, we sort of chose something sort of between here and here that should be moving to give it enough sampling space to move that entire loop out of the way. So as you're coming up towards, for example, if there's a helix coming up, you obviously wouldn't move that, shift that helix, but we have quite a lot of scope of movement here. But if we move it from about here to about here, that should actually be able to pull that whole loop out of the way. So you can do that just by hovering over things, and you can see the numbers here in the bottom of the Maestro interface are about 442, 434, something like that will allow you to put something into this panel. So if you just highlight also the loops here, just if you randomly go through and highlight things, you'll see which parts of the loops is now relevant to the thing that you're trying to move. If you want to edit the numbers, you would just go ahead and edit the numbers here. So 437 to maybe a smaller part, say 443. And then if you highlight that part of it, you can just see it's that section of that entire loop that could be. So there are a number of options and constraints that you can put in that I won't talk about, but in general, this is how you would run it. You can also, go to our manuals to get more details. And the kind of output that you would get from something like this is, because this is not running a induced fit docking, it's running a prime loop refinement, you can kind of see the output structures here, but you're obviously looking for a prime energy in this case, and the lower the better. So that's the kind of movement that we're getting from that ligand. And what we're trying to do is make sure that that pace selective ligand can actually sit in a place where that loop is actually moved out of the way. So that's how we would make a decision as to which loop to keep. If you wanted to run this entire workflow through induced fit docking, you can certainly do that. So let's just go back to the slides. We've already seen how you would set things up through the induced fit docking interface. So you'd work your way through this entire thing. But for the loop modeling part, you would simply run that out and write it out to a command line, because that's where the loop modeling section sits. It's not, unfortunately, embedded into the GUI. But you set the protocol up, write out the file for editing the loop section in the input file. So you see here a little cogwheel. If you click on that, you can write things out, and that creates an input file. And if you look at that input file, the top section of it will be about the prime loop prediction. The rest of it will be exactly the same as the way that the GUI is laid out. So it will be initial glide docking stage, it will be setting up the glide docking and then going through the prime part for the refinement of the residues. But in addition, we've got a loop section here that's not in the GUI. So here we would simply put the start residue and the end residue number. There's an energy number, there's an energy sort of window that it saves structures in, or you can change either of these two. And you would have to comment out this section. Okay, so if I were to write that thing out from the IFD input, I would start to fill this all in as before, and then go ahead and write out the file from here. And once I've written that file out, you can actually see that that's what the file looks like here. And this is the section here that I would start to change. So I could literally just type my new numbers in here. So this is chain A, these are the residues, so 467 I think it was. And then I would just remove all of these hash entries here, save the file, and then run the file using $Schrodinger/IFD and your input file name. And this is the input file here, so that's why I just called it test.imp. And you have some options here about how to run it as well. So if you want more help on this IFD-H for getting more synopsis on how to run that, and that's basically how you would run things in IFD and in extended mode. If you want more information, you can go to our tutorial section inside Maestro and download a tutorial. And just to give you a feeling for the kind of targets that we've tested this on and how we've docked ligands from different structures into the receptor target to get improved RMSDs. So we've gone from 6.5, for example, in the aldose reductase to 0.9. You can sort of quickly benchmark against if you're working on the same kind of projects and proteins, and give a feeling for what you can expect. Well, for now let's take questions. Thank you for your attention, and don't forget that any questions you can go to our help people. You can look at the seminars on our webpage and also go to our knowledge base. So Rita, let's open for questions.